Thank you, everybody. This is John Maslowski with Castle Creek Biosciences, and I'm excited to give you an update and overview of our company and our technology platform. I wanted to thank ARM for having us present today and appreciate them putting together this virtual conference this year, and hoping everyone is safe and looking forward to getting back together in 21 to meet face-to-face. A little bit about our company, just a quick disclaimer on confidentiality. But moving into the introduction. So we are a privately held company that is developing platform of cell and gene therapy products with a focus on cutaneous disorders, particularly skin and other connective tissues, where we could deliver genes of interest and treat rare monogenic disorders in those areas. And we're headquarters in Exton, Pennsylvania, which is outside of the Philadelphia region. So really exciting growth here in our industry. And we are really just moving forward now, trying to expand ourselves into a platform approach. Our lead indication, which is what I'll focus on my presentation today, is D5, which we formally refer to as FCX007, which is currently enrolling in a phase 3 trial. I'm very excited about the progress to that point. And we'll be giving you today some background on the product, a little bit about our manufacturing capabilities and where we are in the trial. We'll also talk a little bit about some of our other capabilities and how we're expanding into new indications, and a little bit about our structure and about our management team, just to give you a snapshot of where we've grown over the last part of the year. So a little on our key investment highlights, and then this will really be a primer of what I'll touch on, talking about ourselves as a leader in a late stage development company. So again, with the lead asset in phase 3, looking at multiple subsets of dystrophic EB, which I'll talk about some of the demonstrated clinical safety and efficacy data we've spoke about in the past, and we'll review some of those again. We believe this is a very attractive commercial opportunity, although rare disease, highly unmet need with a lot of patients who really need a good solution from a genetic standpoint, and we believe we can do that and also deliver in a big way from a commercial opportunity, and also look at the additional platform value. And I'll talk a lot about the autologous fibroblast shortly, which we base our platform on, and what are some of the ways we can expand the utility of that as well. We have validated in-house manufacturing capabilities. I've spoke about those in the past, and we'll review some of what we're doing with our in-house facility and how we can have some expansion possibilities. And of course, we are growing our world-class management team. We've had a lot of hiring this year, a lot in the operational, clinical, regulatory, and just recently brought on a new CTO, who is Dr. Greg McMichael, and very excited to really be moving our platform into the next stage as we go into not only phase 3, but starting to think about what this could look like commercially. So a little snapshot on our pipeline, a little snapshot. As you can see, is our lead indication, which is D5, formerly FCX007. We're giving it, based on its new USAN name, a nickname here of D5. Currently in phase 3, enrolling and looking to have a BLA submission on our target sometime in 2022. We're also looking to expand that indication. We are working in recessive dystrophic EB. We'd like to see that expand into the dominant subtype, which would follow shortly after, so late stage for that as well. We also have another indication. I won't touch on it today due to time, but FCX013 for localized scleroderma. This is now a gene-modified fibroblast that we'll be using to treat this sclerotic disorder. This is actually now in the clinic as well in phase 1. We're very imminently looking to dose our first patient, and we have 4 patients enrolled. So looking to collect some data and move on into the next stage of that in 2021. And with positive outcome, we'll be reporting more about the future of that program as well. We have a lot of exciting possibilities for that particular candidate. We're also starting to think about, now that we're trying to establish a platform, where else can we go with these cells and how to genetically modify them. So we're starting to think about ways of expanding our use of collagen and other collagen disorders. So some new platform opportunities that we'll start exploring and really moving into as this plays out over the year, in VEDS and osteogenesis imperfecta, 2 examples of things that we can move into and starting to do some work on as well. So a little bit about the platform, and then I'll move into the indication. So as I mentioned before, we are working with autologous dermal fibroblasts. So obviously a very highly ubiquitous cell in the skin, easily accessible. So, through a simple biopsy punch, we can access these cells and then use them to expand and culture and genetically modify them with genes of interest. And obviously they're functionally critical for structure of skin and play a big role, for instance, in RDEB and collagen synthesis, which is the core of that particular issue. And we've shown this clinically and in the past in our experience with these cells, very good safety use of these and the ideal vehicle for bringing these to the area of disease. Skin is a very large organ, so to locally deliver these cells to the area of disease, we think the fibroblast makes a lot of sense and have been using that as the core of our platform. As you can see on the right there, autologous fibroblast is the core cell that we use as the transit vehicle, but the lentivirus vector is what we've used to date in order to bring the genes of interest in. And, the fibroblasts we've had a lot of history with, the autologous approach, especially in diseases with high immunogenic concerns like RDEB, reduces rejection rates. We've had this established manufacturing plan for this for over a decade. So we think it was easily translatable into the new indication platform. And as far as lentivirus, the main reason we chose it is these genes are large. So the packaging capacity of lentivirus made a lot of sense. It has integrative nature to it, allows us to look at chronic protein expression. So in a lot of these disorders where we need that long-term sort of protein presence, this really makes a lot of sense beyond that they transpose the binding, non-dividing genes. So in the end, for these particular class of diseases, this combination made a lot of sense. It allows us to redose. Skin is, again, as I mentioned, a large organ, low vascularized, so sometimes these systemic approaches just don't really pan out well. So we can redose. We can treat different areas over time, over the course of the patient's lifetime, and use bank cells in order to go back and treat new locations. So a little bit about the manufacturing process, just very high level. It is autologous. Again, we felt this was the best way to go for these types of indications. We collect a biopsy punch of cells, very, very minimally invasive. Even in highly sensitive skin and Rdeb patients, we've had good success with collecting starter material. We culture them in our GMP facility here in Exton, PA, and then they're introduced to the lentivirus in order to transduce them and bring the gene of interest in. And then what we do is we continue to expand those cells, and the goal there is to create an initial large bank, because we do believe these patients will come back to that bank, and then we'll be able to treat other areas of the body, treat other critical areas as they're coming back for their routine care, which is typically quarterly for Rdeb patients. So we do take about 15 weeks. We're projecting commercially to produce that initial bank, but for every follow-up injection, it's only about a 2-week turnaround to prep cells and ship them, which is about the time it takes to schedule and bring a patient. They don't sort of hop on a plane. They have to bring caregivers and supplies. So we think commercially that will make a lot of sense for the lifestyle of the patients, and we think will fit well in their journey. So again, bank of cells, come back for the patients and treat them over the course of a lifetime. And so what does the future look like? And we've been looking at all sorts of ways to use these cells a lot more broadly, using allogeneic approaches for other indications, if it makes sense there, for high capacity non-viral vectors to look at other collagen-based disorders with larger targets. The 013 program alone I mentioned has a lot of utility from sclerotic disorders, not just in localized scleroderma, but systemic scleroderma or keloids or organ sclerosis or other bone disorders. So we believe that in itself could be its own platform, and excited to see some early data there. And then we have a ongoing library from the past of an autologous product we had approved some years ago of INDs just for other indications in phase 2 that we still own outright, and could always revisit some of those, and looking at ways to differentiate cells through MSC or iPSC pathways in order to target yet additional organ systems. So we think there's a lot of very broad ways we can continue to use this and treat all over the body in different types of gene therapy approaches. So I think we all know, very importantly, the requirements for manufacturing and how a lot of the feedbacks from the agency has been on CMC. So we've put a big investment in making sure we had our own manufacturing capabilities, which we do. We have about 13,000 square feet here in the Philly region. We're manufacturing both products in this area, so it's a multi-use facility. We do have enough capacity here to service the US market for RDB upon launch. So well vetted over history. It's been inspected by the FDA in the past. And so we're doing that. We have all these ongoing process improvements, and not only in the facility, but ways to improve the process even further prior to BLA, and looking at e-systems to help streamline the commercialization of this as well. And also are working with a CDMO to handle a lot of our vector manufacturing for now, which we will look to bring in-house in the future, but managing with a great partner who has had a lot of late stage experience, so we're covered from end to end. So just briefly on RDEB, I'd like to talk about the indication. I'll move through. It is a rare skin blistering disorder that comes from a mutation in the COL7 gene, which produces a protein called type 7 collagen, which binds the layer of skin together. So think of it like Velcro, the hoops and the lattice underneath. The hoops would be sort of like an anchoring fibril that is formed by collagen 7. And if missing, skin layer floats on one another, it blisters, it then breaks, it creates chronic wounds, and it just creates this lifetime of an awful disease with pain, all sorts of different manifestations. And then what becomes highly problematic is mortality from squamous cell carcinoma and eventually and sepsis as well. So obviously, even though a skin disorder, highly impactful on the patient's lives and actually very expensive to care for, a lot of home care to do that. And so we've developed FCX007, now called DeFi, just to address that. It is a genetically modified fibroblast that's encoded with the COL71 gene in order to locally deliver it, produce a protein, and create a wound healing situation to improve the condition. This has been in development for several years now. We work very closely with FDA. It has RMAT designations along with fast track orphan, rare pediatric disease designation. And then we've also received some grant money from the orphan decision of FDA to develop this, and had an end of phase 2 meeting in 2019. So a lot of great progress, a lot of good work with the FDA in order to get this far. And again, we locally apply this, have it produce a protein, and have tested this in a phase 1/2 trial, which I'll talk a little bit about the data from. So back in January, we presented a poster at EV 2020 in London, looking at 6 patients with 10 wounds that were treated, followed out to a year with no serious adverse events or product-related adverse events, no RCL detected, or no autoantibody, very importantly, detected locally or systemically. And as you can see, one of the primary endpoints we're centering on is complete wound healing. And as you see below, at complete wound healing, out of the 10 wounds that were treated, we saw 8 of them completely heal by 12 weeks. And we did this as an intra-patient design. The matched untreated wounds were unhealed. And we saw the trend below, as you can see, continue 25 weeks below. So impressive wound healing results so far, which led us to that end of phase 2 meeting. And a little bit of pharmacology as well, as you can see, as far as 52 weeks, and then patient B on the right, 32 weeks, we saw COL7 expression in the basement membrane zone post administration. You can see from 4 to 32 weeks, actual accumulation of collagen 7. So pharmacology does suggest we have the wound healing endpoint as well, which led us to the DeFi RDEB phase 3 study. This is a study that's currently recruiting, and you have the NCT number below. We are looking at chronic wounds that are matched to a untreated wound that will be dosed, looking at a primary endpoint at 24 weeks of complete wound healing in proportion to the non-treated wounds. So intra-patient design, open label, and we're looking at this in 24 patients, and we are currently in 3 sites and looking to expand from there, and look at some secondary endpoints as well, 12 weeks, and some pain outcome measures. So very excited to see this running, and really we'll be happy to report about some of our advancements over the year, and excited to really get this enrolled and see the data. And in the end, we think we have distinct advantages with this product. It's, these autologous fibroblasts have had a lot of clinical experience, at least in our hands, and including an approved product we had back in 2011. And also Lenti, as everyone knows, has gotten quite ubiquitous, become quite ubiquitous through a lot of clinical trials. And as you know, even in approved products, including Kymriah, So we think that the regulatory uptake of this is quite understood, and we see that with the agency ourselves. We believe the integrated approach is really important for these diseases, and we think we have a very nice approach to do this and create a longer term expression so it doesn't burden the patient as much with returns, including our every 12-week injection. We also looked at this in chronic non-healing wounds, and which we know are the highest, most impactful. All of our data are in non-healing wounds. And in the future, we'd love to see this used in intact skin. Clearly, our current indication is testing in wounds. So, that could be the future where we try to do this more in a preventative fashion rather than trying to treat the problem. But again, as with clinical development, we'll start where we have some measurable devices and go from there. So with that, that is the end of the presentation. Again, we're very excited to see our progress. We're happy with our situation and well capitalized through, and very excited for the 013 and future expansions as well as we see this platform grow. And we'll be looking forward to providing future updates. Thank you very much.